Today we're going to look at the Huberman Lab podcast, episode 16, How Hormones Control Hunger, Eating, and Satiety. The key ideas discussed in this episode are: Number 1. Biology and physiology of appetite. Number 2. Damaging effects of processed foods. Number 3. Insulin. Number 4. Breakdown of cholesterol. Number 5. Supplements and prescription drugs. Idea number 1. Biology and physiology of appetite. The hypothalamus is a control station for hunger, eating, and satiety. It is involved in promoting both feeding and not eating. The arcuate nucleus is a set of neurons in the hypothalamus. The melanocyte-stimulating hormone MSH is released from the pituitary gland and reduces appetite. It is activated by ultraviolet light to the eyes and keeps appetite in check. MSH is the reason animals and humans tend to have reduced appetite in the warmer months. Agouti-related peptide AGRP. These neurons stimulate eating and emotion involved in anticipation of food. If stimulated, you will eat like crazy. Ghrelin. Ghrelin is released from the GI tract and increases the desire to eat by stimulating neurons that increase hunger and anticipatory signals. Ghrelin causes cravings or desires to eat certain foods at certain times of day. If you generally eat at the same time daily and begin feeling hungry at those times, it's ghrelin, which activates the sensation of hunger. If you want to change the eating window, ghrelin secretion can be shifted by 45 minutes per day, so ease into it and push back the window every day until it's where you want it to be. Cholecystokinin, CCK. It is released by the gut and has a powerful effect in reducing appetite. CCK release is governed by neurons and mucosa of the gut microbiome and stimulated by fatty acids, specifically omega-3s, amino acids, and specifically glutamine and sugar. Glutamine can reduce sugar cravings but also increase blood sugar levels. Many cancers and tumors feed on glutamine, so always talk to a doctor before supplementing. Insular cortex gets input from the mouth and has powerful control in whether you enjoy what you're eating, the sensation of eating, and whether you want to continue eating, etc. Endocrine signals are involved in hunger and the desire to eat. Idea number 2. Damaging effects of processed foods. A calorie is not just a calorie. Emulsifiers in highly processed foods shut down CCK and turn off recognition of satiety. Emulsifiers in highly processed foods limit the gut's ability to identify what is in the food and send satiety signals to shut down hunger. Neurons in the gut that sense sugar trigger the release of dopamine and encourage you to eat more sugar. Highly processed foods increase weight gain, disrupt lining of gut, and disrupt hormonal and neural systems that increase desire to eat. Increased consumption of highly processed foods has caused staggering increase in diabetes, obesity, and other related diseases. Idea number 3. Insulin. Food is broken down into sugar and released as energy. Blood sugar needs to be kept in at a certain range. If glucose gets too high, neurons can be damaged or killed, causing loss of sensation in hands, feet, and eyes. Type 2 diabetes. Insulin is secreted from the pancreas, but the person is insulin insensitive and is often associated with overweight and obesity. Type 1 diabetes can often be managed by diet and lifestyle. In type 1 diabetes, the pancreas produces little or no insulin and is often associated with weight loss. Macronutrients are associated with an increase in blood glucose in the following order from highest to lowest: carbohydrates, then fat, then protein. Glucagon is secreted when hungry. The insulin system manages glucose and the glucagon system. The glucagon system pulls energy out of the liver and muscles for fuel until glycogen stores are depleted. After they are depleted, then glucagon pulls from fat. The order you consume macronutrients influences blood glucose levels. Carbs and fat early in meal give rise to blood glucose. Eating fibrous vegetables first will blunt glucose increase. Blood glucose levels can be heavily modulated by moving and exercising before eating. Zone 2 cardio that lasts 30 to 60 minutes dramatically increases insulin sensitivity and stabilizes blood sugar. High-intensity interval training and weight training repackage glycogen and trigger mechanisms to shuttle glucose to glycogen and storage of food. Idea number 4. Breakdown of cholesterol. You always want LDL to be low and HDL to be high. Fats don't like water, but you need to move fats in the bloodstream, so HDL and LDL allow them to move through the bloodstream. Adequate HDL is good because you can move fats to tissues that manufacture hormones. When LDL is high, you can get a buildup of fat. There's a lot of debate about how dietary cholesterol impacts LDL and HDL. How to keep LDL, HDL in the proper ratio? Manage glucose. Idea number 5. Supplements and prescription drugs. Metformin reduces blood glucose by changing mitochondrial action in the liver. Berberine is over the counter and mimics metformin with a similar pathway and results. Chromium, ginseng, magnesium, apple cider vinegar, and acidic foods have been shown to have a minor impact on blood glucose. Caffeine has been shown to significantly increase blood glucose. Stevia is the best in terms of sweeteners and will not raise blood glucose like other artificial sweeteners, but the sweet taste itself will increase the desire to eat, so will probably cancel out. Yerba mate can help regulate blood sugar and has a slight appetite suppressant quality. This is the end. Thank you.